A little pause before we get into some speakers just to talk about how far we've come. And so this is the seventh annual research symposium. And so this is a record year with over 70 submissions. So to those who either submitted or to those who have encouraged your trainees, faculty, students, we hope to keep growing this every year. We'll show the faces of the 21 scientific reviewers. Every single one of the abstracts had at least 2 people review them, if not more, for the typical criteria of significance, innovation. And so, want to give a round of applause to everybody that took the time. Of course, this is uncompensated for reviewing all of these, so we can thank these people. And if you go to the next slide, so of the 70 plus, there was about 62 that were invited, that you can see posters were invited here today. And of those, there was 11. The 11 top scoring abstract winners are shown here. So I won't go through reading each of their names, but please take a moment to see the breadth and depth that this spans from AI to basic science, all different areas of the body. So again, let's give a round of applause to all of our abstract winners. And I believe, that afterwards, for those 11, please find Yi or someone from the R&E team. I think there's a certificate and gift card. I don't want to overpromise, and then there's no gift. Gift card and a certificate. So congrats. And then the top scoring is here, Divya. So she's an undergraduate student in the Department of Biology in Dr. Timothy Mead's lab. And so she'll be speaking to us. So that part of being the top abstract winner, they get to present to us. So this is pretty incredible. And it's about you gotta pronounce this for me. Is it geophysic dysplasia, which I had to look up. This is like a step 1 board question. And so I'm super excited to learn about this and what you're gonna be presenting today. So we invite you to come up to the podium. And it's geliophysics. Geliophysics. I'm an expert in it.
Thank you for the opportunity to present our research today. So geliophysic dysplasia is an extremely rare connective tissue disorder with no known cure. It affects approximately 1 in 1 million people, with only 100 cases in literature. And those with geliophysic dysplasia, or GD, typically have a 30% mortality rate by the age of 5. So these images are of a patient with GD, and notice the joint contractures, the delayed bone age, and the shorter skeletal elements. Patients with GD typically also show cardiorespiratory complications like heart valve dysfunction, as well as short stature. So this is a growth chart of patients with GD, and notice how they're all significantly under the 2nd percentile regarding their height. This includes other genes that can lead to GD, like LTBP3 and fibrillin-1, but we are focusing on ADAMTSL2 since most of the cases of GD are due to mutations in this specific gene. So ADAMTSL2 is an extracellular matrix protein involved in cell signaling and growth factors, and it's involved in TGF-beta regulation as well as Wnt signaling. Mutations in ADAMTSL2 can lead to GD1 and connective tissue dysfunction due to its role in connective tissue biology. So now that we know that mutations in ADAMTSL2 can lead to GD1, there is a lack of preclinical models to study this. So we developed a mouse model using a specific patient mutation, the same one I showed you guys earlier, using CRISPR-Cas9. And we changed a guanine to an adenine, and which changed an aspartic acid to an asparagine at position 167. And the genotype distribution at postnatal day 7 shows that there is a reduced percentage of the mutant pups, from an expected 25% using Mendelian genetics to a 14%, which shows the reduced survivability of this mutation. At 2 weeks of age, it is shown that you can see a difference between the mutant mouse compared to the wild type with a significant reduction in the body length as well as weight. Because the mutant mouse is significantly smaller than the wild type, we decided to look at the measurements of the skeletal elements using X-ray analysis. And we found that the long bones, metacarpals, and metatarsals were all statistically significantly decreased in length compared to that of the wild type. And we had the same results for the forelimbs. Because of the significant decrease in bone length, we decided to isolate the hind limbs in order to further investigate the cartilage growth plate. And we found that this is the femur at 2 weeks of age, and we stained this with saffron and O to visualize the proteoglycans in the growth plate. And we found that the mutant mouse had significantly less growth plate compared to that of the wild type, and this includes the columnar and hypertrophic zones as well. We found the same thing in the 6-month mice as well. And GD patients also typically show valve regurgitation that you can see in echocardiograms. So we decided to look at the aortic valve, and we found that there was a significant increase in the aortic valve area in the mutant mice compared to that of the wild type, and this can lead to valve regurgitation or secondary heart dysfunction. We found the same thing in the 6-month mice, and because of the enlarged aortic valve areas, we decided to look at the organization of this, and we found that they were considerably disorganized compared to the wild type. So notice the blue proteoglycan layer and the yellow collagen layer are much more organized in the wild type compared to that of the mutant, so they are disorganized. Finally, we decided to look at the lungs because patients with GD also show respiratory complications like tracheal stenosis or tracheal malacia. And we isolated and stained the lungs with hematoxylin and eosin to visualize the bronchioles and the alveoli. And we found that in the mutant mouse, there were much more occlusions of the bronchioles compared to that of the wild type. And this matches the patient phenotype and can lead to early mortality. So now that I've shown you that our mouse is a model for GD, let's talk about some future directions. So the first is the use of single-cell RNA sequencing. We'll use heart valves and embryonic limbs as an unbiased approach to further understand the downstream effects of the loss of ADAM-TSL2. We will also further utilize in vitro studies using known activators and inhibitors, or activators and inhibitors for known interactors, and possibly even correct the mutation using CRISPR-Cas9. And with that, I'd like to thank our collaborator, Dirk Humbacher at Mount Sinai in New York for the mouse model and the x-rays. I'd like to thank Connie and Sophia, whom I've worked very closely with on this project, sectioning, staining, and quantifying all the histological slides. I'd like to thank the rest of the lab and my mentor, Dr. Mead, for the guidance and the opportunity to work on the manuscript. And I'd like to finally thank the funding. Thank you for your attention.
Absolutely fantastic. I get the pleasure. I'm not going to let you guys ask any questions. Our undergraduate student. I'm going to ask one or two for you. So, question, just because this involves the extracellular matrix. Clearly, you're focused on bone. What about the skin?
So because it's involved with the connective tissue and mutations in that lead to connective tissue dysfunction, patients with GD typically do show a thicker skin. And we decided to look at the mouse as well, and we found that there was a difference in the thickness of skin between the mutant and the wild type, at least in this layer. But we do have to take more samples of that to get a more conclusive result.
Fantastic. All right. We'll leave it at one question. That was an amazing talk.